Okay, good afternoon, everyone. So first of all, I would like to say thank you for the organizer that gave me the chance to present my work here. So today I will talk about the molecular mechanism of the temperature-responsive flowering in the model plant Arabidopsis. So flowering time is tightly controlled by the environmental factors such as the photoperiod, temperature, water, while the internal factors such as the circadian clock, sugar, and a soil phytohormone. Our lab mostly focused on the role of the temperature in the flowering time. So if you see here, in the past 50 to 100 years ago, we see a rapid acceleration of the flowering time of this chestnut tree from the late March into the early March or even late February, which coincides with the elevation of the global temperature or climate change. The acceleration of the flowering time by temperature has a devastating impact in our agriculture because it will reduce the yield of our major crop plant. So our lab studied this phenomena using the model plant Arabidopsis. If you see here, the Arabidopsis plant flower late when we grow at low temperature and flower early when we grow the plant at warm temperature. So the question that we asked in our lab is what is the molecular mechanism behind this phenomena? So flowering time itself is controlled by a molecule called Florigen, which actually a protein encoded by the gene called flowering locus T or FT. FT is transcribed in the specific cell in the vascular tissue in the leaves called companion cell. And once it's translated into protein, FT protein will undergo long-distance transport to the shoot apical meristem and induce flowering. FT actually belongs to a conserved family protein called phosphatidylethylamine binding protein or PEPD. However, the ability of FT to interact with phospholipid is still poorly understood. So the question that I asked is whether could FT interact with phospholipid? And if yes, what is the implication to the flowering time? To answer this question, I first do the in vitro assay. And indeed, I found that FT actually interact with the several phospholipids, especially the phosphatidylglycerol or PG. So I confirmed those in vitro data with the in vivo experiment. And indeed, using the co-IP, I found that FT physically interact with the PG in vivo. And when I did the mass spec, I found that the FT prefer to interact to the 34,4 PG species. So from all this data, I conclude that FT indeed could interact with the PG, which is a negatively charged phospholipid. The question is, what is implication in the flowering time, right? So to answer this question, I gather several mutants in the PG biosynthesis pathway, which means that the mutant will have a problem to synthesize PG in plant. And I check the flowering time. If you see here, the wild-type plant flower later at 16 degrees, and the PG deficient mutant flower actually early at 23 degrees, and much more earlier at 16 degrees. And when we calculate the leaf number ratio, we found that the PG deficient mutant actually insensitive to the temperature. So we conclude that PG is actually a floral repressor and it works in the temperature responsive pathway. So how can PG affect the flowering? That's the question. So to answer those questions, we did a membrane fractionation assay and detect the presence of FT in the membrane and also in the cytosol or in the soluble fraction. So interestingly, if you see here, when we grow the plant at low temperature or 16 degrees, we see a higher level of FT in the membrane, coincident with the reduce of FT in the soluble part. However, if we have the mutant that does not produce much PG, in this case PGP1, the level of FT in the membrane is decreased, following by increase of FT in the soluble fraction. So I confirmed this result using a different experiment. This is immunogold data. We see the level of FT in the vesicle is increased when we grow the plant at 16 degrees. When we have the PGP1 mutation, the level of FT in the vesicle is decreased. So using the protoplast system, I add exogenous PG. If you see here in the PGP1 mutant in the mock treatment, there are not much colocalization of FT in the trans-Golgi network, in this case, one of the membrane. But once we add the PG, we see more colocalization of FT in the TGN. These results suggest that FT accumulate in the membrane under low temperature in the PG-dependent manner. So we know FT interact with PG and it affects the intracellular distribution of FT in the cell, but how can it affect the flowering time? To answer those questions, I perform a Cotyledon grafting surgery. So here you just simply cut the cotyledon of one plant and put it in another plant. So the recipient is the FT-10, which is the plant does not produce FT. And if you see here, the plant flower very late. But once we graft with another plant that has functional FT, the flower become early. Here you see I graft the FT-10 with the 35S-FT plant, which have a lot of FT. And surprisingly, if you see here, when we grow the plant at 16 degrees, the plants at 16 degrees still flower later than compared to the plant that grow at 23 degrees. However, if we have PGP1 mutation, the effect of the temperature are gone. This result suggests that at 23 degrees, the FT probably move quite fast, but at 16 degrees, the FT move quite slow. That's why we see a late flowering at 16 degrees. But if we have PGP1 mutation in the system, the FT move as fast as the plant grow at 23 degrees. So, as I said before, FT is expressed in the specific cell called companion cell. So it needs to move from companion cell to the sieve element and then move to the shoot apical meristem. So here I see the distribution of FT in the companion cell and sieve element. So interestingly, if you see the plant that grow at 16 degrees, we see more FT accumulate in the companion cell, while we see less FT in the sieve element. This result suggests that at 16 degrees, we see less movement from CC to the SE. However, if we have PGP1 mutation, we see the number of FT in the companion cell is reduced following by increase of FT in the sieve element, suggesting that in the PGP1 mutant, the effect of temperature of this translocation is gone. So as a summary, what we found is first is FT is actually could interact with phospholipid, in this case PG, and this FT-PG interaction promote the membrane sequestration of FT under low ambient temperature, and this sequestration will prevent the long-distance movement of FT from the companion cell to the sieve element and to the shoot apical meristem. Yeah. So I would like to say thank you for my mentor, Professor Yoon An, and also Dr. Snejana, and also our collaborator and our funding agency. That's all. Thank you.
We have 1 minute delay, so I will get one question from the audience. Then I have one question. So you show that Florigen binds to phosphatidylglycerol specifically, yes. And then you deleted phosphatidylglycerol in your model. And then what's the effect of outcome downstream phospholipid species when you deplete phosphatidylglycerol? Because I think it will not be the end product.
Yes. So in the mutant of the PG mutant, there are several other phospholipids also perturbed. Yeah. So yeah, that might also implicate to the flowering time, but we didn't go that far yet.
Yeah. Okay. Thank you. Thank you for your nice talk. And then